start, so Lynn, I can see that you joined the Zoom call, and we have Stephanie. So that would be the panel. So yeah, welcome everyone to Iftikharul alums pre-completion seminar. Iftikharul also goes by Russell. And so Russell did a Bachelor of Science at University of Chittagong in environmental chemistry, and then Master of Philosophy in Japan at University of Kanazawa, and then joined us here studying plant ecophysiology. And so the title of Russell's presentation is The Importance of Acclimation in Driving Plant Physiological Responses to a Changing Environment. And take it away, Russell.
Welcome everyone. Today I'm going to present my PhD pre-completion seminar where I'll be talking about plants' acclimation and their physiological responses to changing environment. So at first I would like to acknowledge the Australian Aboriginal and Torres Strait Islander people as the traditional owner of the land and water where I live and work. So here, all of us, we know that the climate is changing and there are lots of environmental stress on plants. So some of the stresses are soil nutrient, which is being affected by different kinds of land use, climate change, global warming. And then we've got drought. So drought is a prolonged dry period where the amount of water is insufficient for the ecological demand of an ecosystem. And then we got heat stress over the last century. We can say we have evidence that the temperature is increasing, and that is causing some stress on the plants. And elevated carbon dioxide. So since the beginning of industrial revolution, the CO2, carbon dioxide in the atmosphere is increasing by a tremendous number and is increasing. There is no chance or any sign of it to go down. So here we can see that the temperature has increased by 1 degree Celsius since 1970, and then since the last 40 years, 50 years, the carbon dioxide concentration in the environment has increased by 100 ppm parts per million, so which is almost 33% since before that period. So how plants respond in this kind of environmental stresses? So they change their leaf morphology to acclimate to those changes. So here is an example where plant from same species grown at different temperatures shows different kind of leaf morphology. And then they close their stomata to acclimate to the changes. So when they have less water, they're facing drought, they close their stomata to reduce the water loss. They alter their photosynthetic pattern, photosynthetic behavior. So in my first chapter, I'll be talking about how plants acclimate to those environmental changes and change their physiological responses. So in this chapter, I did literature review. I identified some environmental stress and how plants respond to those environmental stress, how they acclimate and adapt over the time, and why Australia is unique in floral diversity and how the plants in Australia are being affected by those environmental stress. And then I identified some knowledge gap, depending on which I made my research objectives. So my first objectives discuss about the water use efficiency of plants in Australia and how the water use efficiency is modulated by precipitation and soil nutrient. And then I tried to look at how the response is adjusted based on their adaptation or their rainfall region. So in this chapter, I talked about the soil nutrient deficiency. And then in the next objective, I try to look at how the plants change their physiological response for environmental stress, drought, and elevated temperature. And the last one, where I try to look how elevated carbon dioxide in the atmosphere is making the plant to acclimate. So here I discussed about angiosperm and conifer. So we have evidence that angiosperms has adapted over evolutionary periods to change of environment, change of environmental condition since the late Cretaceous period. So my thesis is structured in 5 different chapters, where chapter 1 is the literature review and where I try to identify the knowledge gap. In chapter 2, I try to address my first objective, chapter 3, second objective, chapter 4, third objective, and at the chapter 5, I concluded with a general discussion. So in chapter 2, I talked about how soil phosphorus drives the divergent pattern in intrinsic water use efficiency of plant across Australia. So before starting my result, I want to show some background discussion about how plant water use efficiency, what is plant's water use efficiency and how does it work in leaf scale. So here we can see it's a cross section of a leaf where through the stomata, plants are uptaking carbon dioxide and at the same time they're losing water. So through the same aperture, the CO2 is going inside for photosynthesis, but at the same time plant is losing water. So they have to optimize their stomatal behavior. So if we consider photosynthesis, photosynthesis is a function of stomatal conductance and that amount of carbon dioxide in the atmosphere and amount of carbon dioxide inside the leaf, which is CI. And if we consider transpiration, which is the water loss behavior of plant, so here we can see that transpiration is equal to stomatal conductance to water, and then the amount of water vapor inside the leaf and amount of water vapor in the atmosphere. So from these two equations, we can derive our water use efficiency for plant, which is the amount of carbon production by the plant for 1 unit of water loss. So here we can see the amount of carbon dioxide in the atmosphere and the amount of carbon dioxide inside the leaf is the main function, and then there is water vapor pressure deficit. So we know that carbon has mainly three isotopes in the nature. So among those isotopes, only 1% is delta 30 carbon 13C isotope, which is the 13th isotope of carbon. So plant discriminate for this delta 13C, and when they're doing photosynthesis, the amount of delta 13C, the amount of 13 carbon inside the leaf and in the atmosphere is discriminated, and with this equation, we can calculate the discrimination. So here we can see that this term CI over CA is similar in both equation, like in water use efficiency and delta 13C. So in my next slide, I'll be using delta 13C as a proxy for water use efficiency for plant. So how do the stomata behave in water stress? So when there is more water, the plant is well watered, then it behaves like this. So the stomata remains open, the carbon dioxide is going inside the leaf, at the same time, some water is coming out. But when there is drought or water stress, plant close their stomata, and that time they can't close it completely, so there is some leakage. And through that leakage, some water is coming out, and a little amount of carbon dioxide is going inside. So to discriminate against carbon 13C, we can see on the left side of the figure, this one, in well water condition, the discrimination is higher for the carbon dioxide in the atmosphere and inside the leaf. So we get higher delta 13C value. And during the drought stress, the plant is trying to close their stomata, but still there is some absorption of CO2 from the atmosphere, but the discrimination is less. So we can say that delta 13C decreases during drought and increased water use efficiency. The plant increased their water use efficiency through less water loss through the stomata. After I started my PhD, I went back to visit my family and got stuck during the COVID. So that time we had to modify our thesis plan, and we decided to use some of the sample we have in hand. So, and study about different transect all over Australia. So over the last 30 years, there have been several transect studied across Australia where people has collected sample from different rainfall gradient. Here we can see in the figure, we can see that those transects. So in 2017, some of my colleagues has collected some sample along these transects, and we had those sample. I analyzed those sample and used those data for my study. And here I'm going to present that study. In this graph, we can see transect study from two different transect, which is these two, the southeastern Queensland transect, which is A, and the Northern Territory transect, which is B. So in this transect, we can see that with the reduction of mean annual precipitation, so if it goes down, the water use efficiency gets higher, and with the increase of, so we can say that with the increase of mean annual precipitation, plant is getting more water, so they don't have to preserve their water, and they're losing more water. But if the water is less, they're losing less water, and they're acclimating to that condition. But the interesting thing about the Northern Territory transect, B, we can see that for a long range, the plant is not changing their water use efficiency behavior much, but at very low mean annual precipitation, it increases their water use efficiency. And we found a similar trend for most of the Western Australia and Northern Territory transect. But for the Eastern Australian transect, like the Queensland one, Queensland transect, Victoria transect, and the transect I'm studying, we found different trend, which is similar to A. So this result shows the relationship between leaf delta 13C value and mean annual precipitation for my transect, which follows the similar pattern to the South Queensland transect. So what's happening and why this water use efficiency is being different for different transect? So we tried to identify, I tried to identify that change of behavior in water use efficiency, and I tried to look at different climatic variable, which is mean annual precipitation and precipitation seasonality, aridity index, which I didn't show here. And then I looked at some soil nutrient parameters like total soil nitrogen and total soil phosphorus. So here I showed roughly the transects, location of the transect on the Australian map. And here we can see that if we consider the mean annual precipitation, which is panel A, So all of the transect has crossed, have a good gradient of rainfall. If we look at the precipitation seasonality gradient, the gradient is not that evident, and also previously researchers have found that the precipitation seasonality relationship with water use efficiency is not well defined. So now I try to look at what's happening with soil nutrients, whether they're trying to adjust the water use efficiency behavior for the plants. So if we look at soil nitrogen map, which is panel C, we can see that the Eastern Australian transect, they have higher soil nitrogen compared to the Northern Territory and Western Australian transect. And for the phosphorus data, we see the similar kind of pattern. So here I would like to mention, I collected this graded data for soil nutrient from CSIR dataset, and for the mean annual precipitation and precipitation seasonality, I got it from BioClimate dataset. So my question I want to answer in this chapter using this transect, I would like to look at how water use efficiency of plant across Australia are being modulated by precipitation and soil nutrient, and how this response is adjusted by their inherent difference in species adaptation. So for that, I used those samples, I analyzed those samples, then I did some data mining from the literature. I used mixed effect model to see what's happening. Then I used two different common garden experiment where results from two different common garden experiments were compared with my result. And then later I explored the gas exchange parameter for Australian plants through an existing dataset. So let me talk about how soil and leaf nutrient are working in those different transects. Here I talked about the total nitrogen and total phosphorus of soil in context of Northern Territory transect and my transect, which is North Queensland transect. So at the first box plot, we can see that the difference is very minimum between Northern Territory transect and North Queensland transect for soil nitrogen, but for soil phosphorus, the Northern Queensland transect is higher than the Northern Territory transect, which is evident in the map also with the graded data. So we can say that North Queensland transect shows higher total soil phosphorus. So now let's see how this soil nutrient value is affecting the plant leaf nutrient. Interestingly, they show almost similar pattern to the soil nutrient value. So the leaf nitrogen value shows no significant difference between them for the true transect, but for leaf phosphorus, they show significant difference, where Northern Queensland values are higher than Northern Territory. Now I tried to check what's happening with this soil nutrient, and for that I used partial plot where these are the partial plot where let me explain what is done here. So in the graph A, I talked about leaf phosphorus, leaf nutrient as well as leaf phosphorus compared with leaf delta 13C, which is a proxy for water use efficiency. And in this panel, in this partial plot, the leaf nitrogen value was put constant, and it was compared against delta 13C was compared against leaf phosphorus. So here for North Queensland transect, we see no difference, no relationship between, no significant difference between leaf delta 13C, which is water use efficiency, and leaf phosphorus. But for leaf nitrogen, we see significant relationship between leaf delta 13C and leaf nitrogen, or my transit, northern Queensland one. But if we consider the Northern Territory transit, we see opposite trend, where leaf phosphorus shows significant significant relationship, but leaf nitrogen doesn't show any significance. So why is this happening? Now, I tried to discuss about taking the whole transit as a single relationship to soil phosphorus, soil nitrogen, and mean precipitation seasonality for the whole transit. And I found that only soil phosphorus shows significant relationship for the transit average values, but soil nitrogen and precipitation seasonality doesn't show any significant relationship. I mentioned about two common garden experiment before. So in those two common garden experiment, seedlings were collected from New South Wales, where the area is high in phosphorus, and in another common garden experiment, seedlings were collected from Northern Territory, where the soil is deficient. There is deficiency of soil phosphorus. And if we compare these two common garden experiments, we can see that leaf delta 13C shows completely opposite relationship with mean annual precipitation. For New South Wales species, here I would like to mention that in the common garden experiment, it was done in plantation or in shed house, where the plants were well watered and the condition was similar. So for the New South Wales species, we can see that with decreasing mean annual precipitation of the species of origin from where the seeds were collected, it shows that with the decrease of mean annual precipitation, the water use efficiency is increasing. And for Northern Territory species, with the decrease of mean annual precipitation of the species of origin, the water use efficiency is decreasing for Northern Territory species. So why it is happening? It's showing different pattern than the transit values. So my assumption is that the plant in Northern Territory, they are adapted to low phosphorus and less water. So they're trying to get more phosphorus from the soil, and for that they're doing more transpiration. They're losing more water, and when the water is getting less, so they're behaving like their original condition. So in summary, in this chapter, I found that water use efficiency shows divergent sensitivity to regional precipitation, and the regional soil phosphorus modulate this sensitivity, and which is influenced by the intercellular to ambient carbon dioxide concentration, and stomata is behind this influence. In the next chapter, I talked about plants' respiration and minimum conductance to drought and heat stress. What is minimum conductance? So leaf, when there is less water, plant try to close their stomata, and then the minimum amount of water loss through stomata and cuticle is known as minimum conductance. So this is the equation for minimum conductance, general equation, where through the small opening of stomata, there are residual stomatal conductance and cuticular conductance. So leaf minimum conductance is very critical parameter for drought-induced plant mortality, and the magnitude of minimum conductance depends on the environmental stresses. When there is the conductance is low, plant lower its transpiration, which prevent the excessive loss of water and saves the plant from hydraulic failure, avoid photosynthetic damage. A recent study done by a mechanistic hydraulic model shows that during future drought, uh, if we consider a constant mean leaf minimum conductance, the mortality will increase by 20% by 2100. And if we consider leaf's minimum conductance as a function of temperature, then it increases by 40%, which is like 60% plant mortality will be happening by the end of this century. So now my question is, how do plants from different rainfall region physiologically respond to drought and elevated temperature? For that, I chose 4 species. Here we can see the occurrence map of those species according to the same symbol. So I choose Eucalyptus pallida, Coriander clapsonia, Eucalyptus exarta, and Eucalyptus camaldulensis. For this experiment, I used 2 glasshouse chamber, where one glasshouse was set at ambient temperature and another one was set at 5 degrees Celsius higher than the ambient temperature. So this study was done at 2 different stages. First stage was well watered, where plants were well watered for a period of time, and at the end of the stage, we measured those plants for minimum conductance and drought respiration. And at the second stage, we induced drought and waited to see whether the plant is showing any drought stress. And when there was some indication of drought stress, then we took the second stage of measurement. So here you can see the glasshouse and the measurement of leaf minimum conductance. So until very recently, it was really hard to estimate the leaf minimum conductance accurately and also the cuticular conductance. But recently, there has been some research published by Diego and his colleagues, where they developed a new technique using the red light to estimate the leaf minimum conductance. I used that technique in my study. So we can see that plant respiration acclimate to elevated temperature. So in my 2 chamber, plant from 2 chambers, the ambient and elevated temperature chamber, the plants from elevated temperature chamber reduce their respiration, which is 0.1 micromole per meter square per second rate. And the respiration rate was different for species, and there was no difference found between the water stress condition. So the plant which were well watered and drought stressed didn't show statistically any significant difference. What's happening with the minimum conductance now? We found that minimum conductance is also acclimated with the increasing temperature. So the plants from elevated temperature shows reduction in their minimum conductance. They're trying to lose less water, and then they're trying to reserve their energy. As mentioned before, the minimum conductance is the residual stomatal conductance plus cuticular conductance. So what's happening with the cuticular conductance? We found that only for Corymbia clarksonia, the amount of cuticular conductance is almost same as minimum conductance. That means they were able to close their stomata more efficiently than other species, and the water loss through cuticle was their minimum conductance. But for the other species, their residual stomatal conductance was higher than cuticular conductance. And the species didn't show any difference for cuticular conductance. In summary, what we found. So we found that plant respiration acclimated to elevated temperature. There was difference between species. Minimum conductance also showed the similar pattern. Cuticular conductance didn't show any difference between species. Till now, we have discussed about how plants are working. Like in the first chapter, I discussed about water use efficiency, which is the how they're trying to conserve their water. Then I discussed about drought respiration and minimum conductance, which is not being mentioned or cannot be identified by delta frequency values. So now I am trying to look at how the evolutionary change of plants' functional group, angiosperm and conifers, are acclimating their responses to increase carbon dioxide. Since the late Cretaceous period, the angiosperm has increased a lot as a terrestrial plant, and the conifers are competing with them, but angiosperm win the competition by a really great number. And there are different diversification in the angiosperm which help them to get the control over the terrestrial plant species. And we can see that with the decrease of temperature, angiosperms are increasing. My question was, how do plant current rate determine their acclimation potential at elevated carbon dioxide for angiosperm and conifers? For this experiment, I chose 4 angiosperm species, 5 conifer species, which are endemic to Australia and nearby region. I did a glasshouse experiment. I used 3 glasshouse chamber where different carbon dioxide concentration was used for growing those plants to give them a long-term CO2 exposure. And at the end, we did I take some gas exchange measurement. And later, for short-term exposure, the same plants were measured at the opposite concentration of carbon dioxide. So here you can see the plants and their measurement being taken at the glasshouse. So how plants are reacting, acclimating at short-term and long-term effect of carbon dioxide on photosynthesis. At the first graph on left side, we can see that with the increase of carbon dioxide, the plants are increasing their photosynthesis, but angiosperms are showing higher photosynthesis, conifers are lower. It's increasing at increasing rate. Other researchers has also found a similar pattern, but what's happening in this graph and B. So here, the plants, the measurement is done at their growth carbon dioxide concentration, and they were grown there for long term. So they had long-term exposure of CO2. Interestingly, here we can see that the plants grown at higher CO2 concentration shows lower photosynthesis. They adapted or they acclimated to that condition, but conifers doesn't show much difference. To check why it's happening, I tried to look at the photosynthetic capacity of angiosperm and conifer, where I used the lowest carbon dioxide concentration and highest carbon dioxide concentration to check the difference between angiosperm and conifer. At higher carbon dioxide concentration, angiosperm decreased their photosynthetic capacity and conifers were unchanged. And the pattern is like this if we want to see what's happening with stomata and photosynthetic capacity. So angiosperms, they downregulate their photosynthetic capacity, but conifers are not doing much, and we can see clear difference between angiosperm and conifers here in this graph. And conifers are continuing to maintain their higher photosynthetic capacity at lower stomata conductance. So from this chapter, I found that at higher growth CO2 concentration, photosynthesis is decreasing at angiosperm, but conifer doesn't show any difference. At short-term increase of carbon dioxide, both angiosperm and conifers are showing increasing trend in photosynthesis. And at higher carbon dioxide concentration, angiosperm downregulate their photosynthetic capacity, conifers were unchanged. So in the last chapter, I discussed about what I have found overall right till now, and that is, if we consider water use efficiency, soil phosphorus is is modulating that behavior along with the precipitation, and this is regulated by leaf stomatal conductance. In the next chapter, I found species from different rainfall region acclimated to their respiration, to elevated temperature and water stress, and their minimum conductance was also acclimated. And in the last chapter, to see the effect of elevated CO2, we found that plants showed different pattern, like angiosperm and conifers, they showed different pattern for long-term and short-term exposure of carbon dioxide. And angiosperm were able to downregulate their photosynthetic capacity, but conifers didn't show much difference. So this is my thesis submission plan, where I'm showing how far I have been with my thesis and how long will it take to me to complete and submit final thesis. I would like to thank all of my teachers, supervisor, my friends, family who have been helping me through my journey. Thank you.
Great. Thanks. Thank you very much, Russell. Do we have questions for Russell? We could start with questions on Zoom. I think you can just speak and we should hear you.
Hi. Hi, Russell.
Hi,
That's a huge improvement. Very nice to see. Much easier to follow and understand. So thank you so much.
thanks to you.
Well, not entirely. But anyway, could you just go to that the last little section? And because I didn't follow, in particular, your very last graph. It looked to me like angiosperms and gymnosperms are on the same. They're on the same trajectory. They're just different amounts of operating G, whatever those are.
So in this graph, for conifers, the blue dots, and then just for red dots. So for conifers, at lower operating stomatal conductance, they're showing higher stomatal photosynthetic capacity, which is the maximum rate of carboxylation.
But they're both doing that. It's just that the relative amount. So there's no difference between what conifers are doing and what angiosperms are doing. They're just a different place on the line.
I have another graph to show support that idea. I didn't show it. So here is showing the stomatal sensitivity to carbon dioxide. How the stomatas are sensitive to the carbon dioxide.
Yeah.
And on this graph, the plants form.
So angiosperms are lower and conifers are higher, but they're still the same. The relationship's the same, isn't it?
But they're more sensitive to carbon dioxide.
Angiosperms are, or conifers are.
Angiosperms are.
Yeah. But it's the same. It's just that they use up more oxygen. What do they have? Fatter stomata or skinnier stomata or bigger stomata or something? Is that what's happening? Like, is it just a physiological difference or a physical difference between the plants?
Yeah, it depends on like the amount of stomata they have and the closure and regulation and how they're controlling their stomata, and all those kind of features are combined and it's acting.
I guess I'm trying to understand your point. Are you saying that conifers and angiosperms are different from each other or the same?
They're different.
But they're not. They're the same. So if you go to your last graph again, it shows you that they're the same. They're just on a different place on the same line. I don't know. Maybe I'm misunderstanding the message.
So the points they're trying to adjust their photosynthetic capacity, and the line is showing the adjustment where they're trying to adjust.
Yeah. So angiosperms are better at adjusting. Like they adjust. Well, they're not even better. It's just exactly the same. They're on exactly the same line. Or are they on different lines and you put one line through them and it makes me think. You see what I mean about them being on the same line? The relationship between their stomatal conductance and their Vmax is identical. They're just one group is higher than the other one, but they're the same. Is that a reasonable conclusion or am I confused?
Yeah, I guess I could step in. So I think that's a good conclusion. And what the experiment showed is that there's this sort of common relationship between photosynthetic capacity and stomatal conductance, and that the angiosperms kind of slide backward on that line in response to high CO2. So their stomata close, they have this more sensitive stomata, and coincident with that, the photosynthetic capacity declines, and so they acclimate in that way, whereas the conifers don't show that acclimation. So when grown at higher CO2, then the two populations kind of come closer together than when at low CO2, if that makes sense, because the angiosperms are.
Yeah,
but as you say, the relationship is a common one that should dictate how the acclimation occurs. Yeah.
Okay, but the angiosperms can slide up and down that line further, is what you're saying.
Yeah, that's right. They seem to be more reactive in that sense. Yeah.
Okay, sorry, Russell, asking hard questions. That was my major question. I'll let it go to people who understand this better than me.
How about to someone who understands this probably worse than you, Lynn? I maybe have just a broader, bigger kind of question, and maybe I missed it early on, but so these angiosperms and conifers, are they native species? Are you sort of looking at sort of showing how native forests might change, or are they wood, forestry or something? What's the?
They're native species, native to mostly Queensland, and southern conifers are like from the southern part of Australia.
Okay, so possibly a broader implication of your work would be seeing how native forests that are dominated by these particular species might fare under kind of higher CO2 or more drought-like, that kind of thing. Is that where it's going? Okay. Yeah, cool. Really interesting. Okay, thank you.
Any questions from the room?
Thank you very much, Russell. This might be in a broader scope of things. When I entered, you were talking about carbon, and you talked about carbon throughout. I am not in that field, but you made me understand it really well. So that was a good way of presenting to me. My question is, now greenhouse gas emissions are major issues globally, and most countries, including Australia, is trying to get to net zero by 2050, starting from carbon neutral to net zero. Forget about the definitions, but net zero means that you're not going to rely heavily on offsets. You are going to rely on natural processes to reduce your carbon emission. And one of the natural processes is through plants or trees or forests. If you are to advise the Australian government how to get massive reduction in carbon emissions through afforestation or plants, what would you tell them? Because you'll be a great consultant for Australian government.
I'll tell them to give me some grant. So yeah, what I found, one of the things where the plant who are adapted already in the region, they are much more able to acclimate to those strains. So it's better to use those native species rather than introducing something else from somewhere else.
Thank you.
I have a question. It's just an interest one. Could you flick back to the slide where you're talking about the gas exchange methods? Or gas exchange measurement methods, sorry.
This one?
No, you referenced the paper that was it a change in methodology in terms of that?
It's a new method.
The new method?
Yeah.
What are the key differences between old methods and the new method?
Previously, there was no definitive method to calculate minimum conductance.
Yeah.
So people were saying that at night-time, nocturnal conductance could be minimum conductance, or when there is a lot of drought, that could be, but they didn't know how to measure it or the exact point of minimum conductance. Yeah. And there was no way to measure in a easy way the cuticular conductance. There was no way to measure cuticular conductance. Yeah. Previous method was you let the leaf dry in a fan for overnight. So when the leaf is dried, then you measure. You assume that plant has lost enough water, it's closing their stomata. What is left is cuticular conductance.
Yeah.
So you're just assuming, and it's taking a lot of time. You're drying the leaf.
Yeah.
But here you can like do it in the alive plant. You just take the plant to the lab, put it under the red light, take the measurement, do some calculation, you can estimate the cuticular conductance.
Yeah. So it's mathematics based calculation.
Yeah.
Thank you.
Mike's got a question. Mike, we can see you have your hand up.
Yeah, a really interesting talk. I guess you guys are getting such amazingly fine-grained understanding of how the different plants work, and you can see across those transects, such a diversity in response and in the trials in the greenhouses. Generally we talk about influencing, global models and stuff like that. Where exactly do you see this level of detail fitting into models?
The model that are present right now, they don't have minimum conductance included much, or even if they have that, they don't have data for at species level. So they're using some constant minimum conductance values for the models, but it's totally different for different species. Here we found that we like in my experiment, I found change. They are different for different species, the minimum conductance. So if we are to include minimum conductance in the model, then it's better to assume what's happening at the species level also, and also like water stress and temperature stress.
So you're thinking that you would map species across the PFT or something like that?
At least for some species, yes.
Yeah, okay. No, cool. Thank you.
Yeah, thanks.
Any other questions at all? Anyone on Zoom or? Okay, please join me in thanking Russell.